Welcome, everyone. Thanks for joining us today. This is the Tools and Technology Seminar Series. For those of you who are not familiar with it, it's basically just a venue to discuss tools, technologies, methodologies in development, already developed, just something maybe that'd be of use, maybe been around for a while, but of interesting use to researchers. So if you're in the room and you have questions, please feel free just to raise your hand. If you're online, please put your questions in the chat box. I'll be monitoring that and can pass those up to Chris. We might save those for the end after he's done with his initial presentation. So today's speaker is Chris Azaldegui, and he is a PhD student in chemical biology.
Great. Hello, everybody. So, like it was mentioned, my name is Chris. I'm a third-year PhD candidate in the Chem Bio program on campus. Also a member of the Biteen lab, and I'm excited to tell you about some pretty awesome work I've been doing imaging bacterial organelles at the single cell and single molecule level. And all of this has been done in collaboration with a postdoc, Dr. Yi Huang, who's a member of the bacterial lab on campus. So to start, I want to give you a brief overview of what the Biteen lab focuses on campus. So we're a very technique-driven laboratory. In particular, we're using single molecule fluorescence. So we're able to detect with high sensitivity single fluorophores. And we are motivated to do this to provide access that ensemble averaging would not do. Meaning that we're able to access heterogeneous dynamics or mixtures to get an idea of what's actually happening at the single molecule level. And another thing that these measurements allow us to do is to implement it as super resolution microscopy, which, if you're familiar at all with the diffraction limit of light, things smaller than around 200 to 300 nanometers will look quite blurry using fluorescence imaging methods. But using single molecule imaging to access super resolution, we get nanometer scale details, as well as being life cell compatible. And then one of our favorite things to do as well is track these single molecules, like I said, to measure their dynamics and get information of the biophysical properties of these individual molecules. And so one of our favorite things to do is to apply these techniques to imaging living microbes. So in the past, we've applied these techniques to measure the starch degradation machinery in our gut microbiome bacteria, as well as building super resolution images of subcellular compartments inside of the cell, bacterial cell. And more recently, we've applied this technology to measuring dynamics of proteins in the nucleus of fission yeast to study epigenetic inheritance. So here, I want to outline my talk by three individual sections here. First, I want to motivate it through the biology that I'm particularly excited by, imaging or understanding subcellular organization via these membraneless organelles. And then next, I'll talk about the work that I'm doing in collaboration with the bacterial lab, doing single cell imaging, applying fluorescence microscopy, as well as my portion doing image analysis to investigate material properties of these membraneless organelles. And finally, get into using super resolution imaging as well as single molecule tracking to study the components inside of these organelles and measure their dynamics. So, like I said, I want to start by motivating this with the biology. And so, we're probably all very familiar with some version of this figure here, where we have a eukaryotic cell that you perhaps seen in your introduction to biology textbooks. And there are very clear features within the cell. One that particularly pulls my eye being the compartmentalization and the significant organization of things inside of the cell. So more strikingly, you have something here like the nucleus that contains the DNA and all the genomic information in the cells, as well as other different organelles. And the reason why we're able to quite readily see these compartments is because they are membrane-bound organelles, which give it a defined physical barrier between the internal components and the external components. However, over the last 15 years or so, there's been a rise in investigations looking at organelles that are not bound by membranes, but rather membraneless, as I'm calling them here. And these are involved in a quite variety of functions, ranging from determining the fate of the cell, as well as involved inside of the nucleus in transcription processes, for example. So not only membrane-bound organelles, but also membraneless organelles are quite prevalent subcellular organization systems. So another example of one of these membraneless organelles are stress granules. And this has been quite, the membraneless organelle field has been of high interest due to observable properties of these organelles using fluorescent cell fluorescence imaging, demonstrating that these organelles are behaving with liquid-like properties, which is kind of going off the status quo in terms of how we understand subcellular organization. So for example, here we have these green foci inside cells that are formed, they're called stress granules, and they are formed after the addition of arsenite. So the cells have a stress response, and they spontaneously create these foci here. And you'll notice here on the right that these foci are quite dynamic and have a particular spherical shape, provided the resolution. And over time, you can see them fuse and then return to this spherical shape nature. And so these liquid-like properties have led to the proposal of a particular mechanism for the formation of these membraneless organelles, being some phase transition or phase separation. And in particular here, I'm showing liquid-liquid phase separation, where you imagine you have a biopolymer, a homogeneous solution of a biopolymer, whether that be protein or nucleic acid, or a combination of the two, that exists in one phase. And as a response to some changing condition, but or a concentration change, the solution can be mixed into two phases of the same components, but one being a highly enriched dense phase, as well as a dilute phase around the organelle or the condensate, as they're termed. So liquid-liquid phase separation, or LLPS, is a proposed mechanism for the formation of membraneless organelles. So far I've introduced organelles, membraneless organelles in eukaryotic systems, but like I mentioned earlier, we're kind of interested in bacteria. And so these membraneless organelles are also present in bacterial cells, and they're quite high importance. So for example, one of the earlier findings of these compartments inside of bacteria was something called a BR body, which is composed of RNA degrading system, so the RNA degradosome, and all these proteins are able to bind RNA, which triggers this phase transition into the BR body, which is a condensate that exists inside of the cell. And so I mentioned earlier that stress can also be a leading factor to components in the cell undergoing a phase transition. And so here we have another proposed system where you get nucleoid compaction. So the DNA inside of the bacterial cell gets compacted due to starvation or other stress responses by this protein DPS, which under wild type conditions becomes highly enriched and creates this liquid crystalline structure. And then, with the model being if there's no DPS present, then we don't get this phase transition inside of the cell. So stress is one way that can lead to phase transitions happening in bacteria, but there are also, these phase transitions also appear to be quite prevalent in just like normal subcellular processes in bacteria. So for example, we have this POP-Z protein, which stands for polar organizing protein Z. And so given its name, it's found at the poles of Caulobacter crescentus cells, which are a model system for asymmetric cell division in bacteria. And so this POP-Z organelle that's located at the poles actually regulates a lot of the phospho signaling pathways that end up deciding the fate of the cells after asymmetric cell division. And so as my COVID project, when the pandemic started, I dove into literature and found a lot of systems where this idea of phase transitions and membraneless organelles were evoked. I'm sorry, I told you about a couple of them, but as you can see here, there's at least 10 and even more now are being, since this was written in around 2020, more examples have popped up. And my favorite one being the carboxysome, and I'm really excited to tell you why, especially since carboxysomes are responsible for around like 35% of carbon fixation. So if you enjoy breathing, you don't have to give thanks to plants necessarily, but rather the carboxysomes, and in particular, the bacteria that contain these carboxysomes. So what are these things, right? So carboxysomes are more commonly termed as bacterial microcompartments, but they're also considered membraneless organelles since they're not bound by a membrane, rather a protein shell. And these are found in cyanobacteria as well as some protobacteria. So on the left here, you can see an EM image of these granule-looking things that are evenly distributed along the cell length. And so carboxysomes, why they're so good at fixing carbon is that they concentrate this highly inefficient Rubisco enzyme with its CO2 substrate to efficiently undergo that enzymatic process. Over almost a decade ago, the biogenesis of carboxysomes was quite of interest, and this paper actually demonstrated how carboxysomes form inside of the cell. So you can see here at the 2-hour point, you get a single carboxysome at the pole, and over time, you start seeing another carboxysome budding off, which if you remember, I told you earlier, the stress granules show fusion events that demonstrate their dynamic liquid-like properties. This may seem a little bit counterintuitive, given that instead of fusing, they're actually defusing or budding off one another. And so there's a reason for this. These compartments actually have a positioning system that allow for this process to happen. And so the characterization of this protein system termed maintenance of carboxysome distribution, and it's composed of 2 proteins, one being A, one being B. This is all work done by our collaborators in the bacterial lab. So briefly, we have this mCherry-McdB protein that from the diagram on the right is a DNA binding protein that is able to form these very pretty oscillation patterns on the nucleoid. So it oscillates back and forth, therefore positioning carboxysomes. However, they themselves cannot interact with carboxysomes, so they have a partner protein, mCherry-McdB, that is localized to carboxysomes itself. And so previous work in the literature studying the internal components of carboxysomes demonstrated that phase transitions may be involved in the biogenesis of carboxysomes. And given that mCherry-McdB shares a lot of the similar properties that many phase transitioning proteins have, the bacterial lab asked the question whether this protein could undergo phase separation in vitro. And indeed, while the mCherry-McdB purified from this S. elongatus cyanobacteria can indeed go LLPS in vitro. As you can see here, you form these lipid droplets in a test tube that are capable of undergoing these dynamic fusion events and ultimately converging to again a spherical shape once fused. And so this activity is particularly interesting because they also found that this phase transition is quite dependent on the pH of the environment where the protein is. And this is interesting because of a proposed model of a pH gradient where the cytoplasm of the cyanobacterial cell is around pH 8, where we would not observe phase transition of mCherry-McdB. However, when you get to the carboxysome internal components, given that the carbonic acid that's present that gets converted to CO2, you see a significant decrease of pH or that's proposed. And so this would correlate well with the conditions where mCherry-McdB will undergo this phase transition, which may point to the phase transition itself having some functional activity in potentially the recognition or the general homeostasis of carboxysomes. So the big question here that we're asking is, can mCherry-McdB undergo phase separation in vivo? And the clicker's not working anymore.
Punching that over to your slide.
Cool. Let's see. Right. Okay, so does mCherry-McdB phase separate in vivo is the big picture question here that we're asking. So next I don't want to stop here and provide an intro to our next section and the experimental methods that we're using to try and answer this question. So in this next section, I'm going to talk about some fluorescence imaging which the data was collected by my collaborator, Dr. Wong. Used to be good. It's just 2 bars. Yeah. I don't know. Okay.
Chris, you might just need to present from the podium.
Okay. We're here. Hopefully everybody can still see and hear me quite well. So earlier I mentioned that there were specific observable properties of these compartments or these membraneless organelles, which suggested that they are behaving as liquid and potentially being formed through this mechanism of phase separation. And so I mentioned that using fluorescence imaging, they were able to observe that these membraneless organelles were forming spherical shaped compartments that were capable of undergoing fusion events, and you can see these things in vitro as well. And because they are behaving like liquid, you imagine that the components inside of these compartments are quite dynamic, and they're able to both internally rearrange as well as exchange with components outside of the dense phase back into the dilute phase and vice versa. And so generally this particular feature has been studied using a technique called fluorescence recovery after photobleaching, or FRAP, where you take your membraneless organelle, for example, pre-bleaching, and you shine a high intensity laser at it to bleach the region here. And if you have dynamic change of these proteins, you expect recovery of fluorescence here. As well as, and again, I also mentioned that these are concentration dependent events. So once you reach a saturation concentration, CSAT, this is where you get the spontaneous demixing. So that's another important feature of these systems. So generally speaking, fluorescence methods provide access for us to try and understand the material properties of these different organelles to see if they are indeed formed through this phase separation process. So Dr. Yi Huang went ahead and took mCherry-McdB and expressed it in E. coli, where we don't have carboxysomes present. And then the motivation with E. coli is that the internal pH of E. coli would be compatible with the pH where mCherry-McdB were to go to space transition, as well as the myriad of molecular biology tools that are accessible to us for using an E. coli model system. So here, when she expresses fluorescently labeled mCherry-McdB, you can see these highly enriched dense foci forming at the poles of the cell. And the reason why they're located at the poles, she found, was that they're actually excluded from the nucleoid, as you can see here through a DAPI stain. Another observable feature is the fusion events of these foci inside of the cell. So here we see a time course of 2 mCherry foci over time fusing into an amorphous compartment. Again, bacteria are very, very small from the scale of a couple micron compared to eukaryotic cells, where the compartments are actually as large as 10 micron. So our resolution here is quite limited. But in conclusion, with this initial preliminary experiments, we found that overexpressed mCherry-McdB forms these nucleoid excluded foci that can fuse in E. coli, giving us a nice model system to further investigate its dynamics. And so here I mentioned we're using mCherry-McdB as a model to try and investigate these phase transitions of bacteria, and the function, generally speaking, of mCherry-McdB is in terms of carboxysome positioning. But we're also using a couple other systems as controls, and both to understand the material properties that they have. So here we have this protein called C1, which I'll just refer to as our aggregating protein that forms these highly dense foci with no fluorescence in the cytoplasm. And as the cells continue to divide, you see that focus remains intact at the same cell fold that it started at. And then finally, earlier I introduced this POP-Z protein that exists in Caulobacter crescentis. And so this protein is the major component of these microdomains, and it only recruits other proteins for the myriad of its functions. So this protein was very well characterized and was actually found. They were able to detect the specific regions inside of this protein that are responsible for this spontaneous assembly, which are these H2, H3, H4 regions. And then this protein also has this intrinsically disordered region, which they found tunes the material state of this compartment, which actually had implications to the fitness of the cell itself. And so because this is quite a tunable system, we're actually using two different lengths of this intrinsically disordered region, one being the 78 amino acid one, which I'll now term the long linker version, which behaves more like a liquid, and then a 6 amino acid version of this, which is somewhere in the middle between a liquid and a solid-like assembly. So to do some fluorescence imaging of these, we switch our fluorescent protein to mCherry to remain consistent, and it would be compatible with other two-color imaging. And so here you see mCDB still forms these highly enriched foci that are denoted here by the white arrows, while also maintaining a significant fraction of a fluorescent signal in the cytoplasm here, which is indicative of this two-phase system where you have a highly enriched dense phase while still maintaining a dilute phase of the system. And so when we look at these two other systems, so the POP-tag SL or short linker, you also get these highly enriched foci forming at the poles of the cells with some fluorescence signal from the cytoplasm. However, with this C1 aggregator protein, you do not, we cannot observe a significant fluorescent signal. So obviously these are all qualitative observations. So we want to be able to quantify this data at the single cell level. So what I'm doing here is taking, segmenting these cells and taking all the pixels inside of them and normalizing to the minimum and the maximum intensity of these and creating a distribution here. So a histogram of the normalized intensity distribution. And so for a homogeneous system where we haven't gotten this spontaneous demixing and cluster formation, you see we have a quite flat distribution. So all the pixels are broadly distributed. But as soon as we start forming a cluster, you get more of the low intensity pixels. So we get an increase here on the left side of the curve. And so these are both mCDB examples, and what we're demonstrating here is that mCDB maintains a solid diffraction in the cytoplasm. Now we take this analysis a little further and apply it to all our systems, our model systems here, and we choose a threshold, which in my case I chose 30% of the maximum intensity, and integrate over the area below that threshold. We get values that are indicative of the ratio and the clustering effect of these cells. So for example, for C1, where we have these very distinct clusters and no signal in the cytoplasm, the integrated value is significantly high, so around 90%. For mCDB, we get a broader distribution, where in some cells we get a really dense cluster with less signal in the cytoplasm, and then a wide spectrum of these happening, likely due to the difference in concentration levels inside of the cell. And then for the POP-tag short linker, like I said, this is behaving something very dynamic, but more like a solid rather than a liquid. So again, you see this highly enriched number of cells around the 90%, but you still get some cells in this lower range here as well. So in conclusion, not all signal of mCherry-McdB is found in these brighter spots, which is indicative of this dilute phase inside of the cell. So next thing that E did was implement this FRAP assay to determine the dynamic exchange and internal rearrangements of these membraneless organelles. So for C1, I introduced this earlier, you have this bright focus, and then so you get a fluorescence intensity that's quite high here, and then you bleach it, so it decreases quite rapidly, and over a period of 300 seconds here, we don't see any recovery here, suggesting that there's not a lot of internal rearrangement or dynamic exchange between the internal and potential external components, if there are any in the system. However, with mCDB, once we bleach here, we do start seeing recovery of signal over time, and the curve will look something like this. So we're currently in the process of repeating these experiments, also applying it to all of the other proteins that we're studying, and doing this quantification. But given this qualitative observation of the pattern here, we can say that there is a significant recovery in the MCDB focused fluorescence over time after photobleaching, suggesting that it's more dynamic compared to the C1 aggregate. So another thing that we're doing, like I said, these droplets form after we reach some concentration, the CSAT. And so what we did here is stop the concentration or the induction of protein after we see droplets forming. And then what we did is trying to change the effective concentration in the system by changing the overall volume of the cells. So here we treated the cells with cephalexin to inhibit division, which would allow the cells to elongate, ultimately increasing the volume and decreasing the concentration of a protein. So what you saw there is these bright focuses ultimately dissolving over time. So you see the signal decrease. For the C1 aggregator, however, after doing the exact same assay, we don't see any differences in the signal of this this focus. And so I can quantify this by detecting and tracking these foci over time and measuring their total intensity. And so here on this plot, that's what I'm showing you here, the normalized total intensity of, in blue, we have these C1 aggregates that over time actually tend to increase in signal, but it's quite stable over a period of 10 hours. However, for this PubTag short linker, you see a slight increase likely due to a delay in the formation of fluorescent proteins after we stop induction. And then beginning around 1 to 2 hour points, we start seeing a decrease in the total fluorescence intensity. And then the gradient of color you see here is representative of the total number of clusters that are present in the assay. So which are also decreasing, suggesting that these foci are dissolving over time. Indicative of this transition once we hit the CSAT point. So we repeat this experiment with mCDB as well, and you can see these foci dissolve over time as the cell volume increases. And here we have the cumulative data of all these 4 different proteins I've introduced so far. C1 remaining a constant, PubTag with the short linker decreasing, as well as mCDB, and then PubTag with the long linker, which is very dynamic and even more so liquid-like, actually fully dissolves within 3 hours of the experiment. And this is just quantification of the lifetimes here of these clusters. So another way we can do this, instead of inhibiting cell division and allowing the volume of the cell to increase, we can actually dilute the overall concentration of the protein by allowing the cells to just behave normally and divide after we stop expression of this protein. So you can see here, we started with these enriched foci, and over time they dissolve and you get a homogeneous signal inside of the cell. For this PubTag phase separated protein, you get the exact same thing happen. And you'll notice that again, we start seeing focus formation once the cell actually clips, and you get a larger effective concentration due to the decrease in overall volume, which is actually quite fascinating behavior here. And then for C1 aggregator protein, even as the cell divides again, you just get inheritance of the polar focus that's there, and nothing really happens. So again, we get the quantification of this. So C1 remains quite constant, and you see that the blue, the shade of blue here remains quite constant, demonstrating that the overall N number of foci remains quite constant, and showing that they're insoluble. And then you see the overall decrease of these 3 other proteins, as well as the full dissolving characteristic with the decreasing number of N, and their lifetimes are shown here. So overall, lowering the protein concentration drives these condensates below the CSAT point, causing them to dissolve and return to the one-phase system where they're fully in this homogeneous region. So finally, I want to talk about the implementation of super resolution imaging as well as single molecule tracking of these specific proteins to get a better idea of the dynamics that are occurring here. And so earlier I introduced these different features that are characteristic of liquid-like compartments. However, given that bacteria are quite tiny, using ensemble fluorescence imaging techniques only provide us a limited view and measurement of what's actually happening here. And so we're implementing these highly sensitive techniques that provide us these nanometer scale details to get a better understanding of the material properties of these proteins. And so how do we achieve this? We have a pretty simple optical instrumentation setup, we have these lasers. Particularly, I want you to pay attention to the 406 and the 561 laser, which we use for our experiments imaging and cherry fluorescent protein, which is in the red. Gets excited with the 561. However, when cherry, when you were too excited, we saw the experiments from earlier. You get all the fluorescence inside of the cell, and we want to achieve single molecule imaging, which is why we use the photoactivative operation here that we can activate using the 406. So this is what I'm talking about here. If you're using just an mCherry protein, you get just ensemble signal, fluorescent signal. And then for the single molecule experiments, we first activate the fluorescent proteins using the 406 nanometer laser. And then we're able to excite these with the 561 and track them and look at them over time, specific single molecules. And then we repeat this over and over again inside of the same cell to then localize and track molecules. And so the spatial and temporal separation of the fluorophores allow us to get single molecule resolution compared to ensemble fluorescence imaging techniques. And so once we have our single molecule data, which here you see what that raw data looks like of a single fluorescent emitter, we're actually able to take the pixel intensities of these. So in here on the X, Y, you should have pixel location, and then in Z, you have the intensity. And you can see it has this, what we call the point spread function, which looks like a mountain. And so we can fit this mountain to a two-dimensional Gaussian distribution to super localize the location of our molecule. And so each molecule is detected with high sensitivity. Using single molecule imaging provides us access to these heterogeneous dynamics and mixtures. And then we're able to pinpoint the very precise location of these molecules and get nanometer scale localization rather than the diffraction limited imaging we were doing earlier. And so these techniques have been already applied in membraneless organelles bacteria, given that they're small. So it makes sense to use these super resolution techniques. So here we have this POP-Z system that I introduced earlier. And so two-color super resolution imaging and single molecule tracking was used to look at the dynamics of client proteins that are able to access this POP-Z microdomain and how they diffuse inside of them. So going from ensemble fluorescence imaging to single molecule sensitivity, as well as these BR bodies, which are present as well in Caulobacter crescentis. So this is actually work done by our lab, where we demonstrated that this RNase E protein, which is highly enriched in the BR bodies, is very has anti-correlative localization with ribosomes inside of the cell. And so to provide you some of the data that I've been collecting and analyzing using super resolution imaging, we have the C1 aggregator protein, which you can, and we have on the y-axis here the level of induction. So we are increasing concentration as we decrease in from top to bottom. And so here you see the C1 aggregator protein forms these highly dense focuses at the pole. And you don't really see any localization density in the cytoplasm, suggesting that all the proteins are being recruited to this polar focus, forming an irreversible aggregate, given all the data that I've already shown you here. Similarly, for POP-TAG, we also get a highly dense focus forming, even at the no induction sample. However, we do see a larger fraction of molecule density in the cytoplasm. And then for the long linker, whenever we don't induce, we see a pretty homogeneous distribution of this protein at the single molecule level, which as we increase concentration, we start seeing these highly enriched regions at the poles, consistent with the space transition happening. And then finally, for MCDB, we get the same effect happening over as we increase overall concentration of the protein. So those were single cell images that I showed you there. But what happens if we take all these localizations over dozens of cells and project them onto one for an average? Again, for MCDB, you can really see the transition here from a homogeneous distribution to a two-state system, getting these highly enriched regions at the poles. And then here's the data for all these other proteins. And then in particular, comparing it to the C1 protein, you get very drastic, what is the word I'm looking for? Contrast between the localization density in these polar foci versus almost no localization density in the cytoplasm here. And so this is indicative of an equilibrium that mCDB has between the mCDB and the condensate versus in the cytoplasmic regions inside of the cell. And so all of this is mCDB localizations, but we're also interested in looking at the dynamics of these proteins. And so here we can take these localizations and then start assembling trajectories of single molecules to get an idea of their motion inside of the cells and how they may be exploring different environments, in particular in these two-phase systems. And so the way we do this is we take a single trajectory and plot the mean square displacement versus time lag. So it's the mean square displacement after delta of t of 1, delta t of 2. And we plot this curve, and we're able to plot this curve or fit this curve to this equation here, where ultimately the parameter we're really interested in is this d parameter, which stands for the diffusion coefficient, which is a representative value of the mobility of our protein. So higher d values means proteins that are quite dynamic and moving rapidly. A low d value is something that's pretty static or moving slowly. And so when we take mCDB in our pre-induction conditions, we're able to take all our d values over all of these trajectories and plot them in log form. And then, so on the x-axis, we have the log of the apparent diffusion constant versus y, just being density. And so you see that we have a high enrichment of fast-moving molecules in our pre-induction conditions and a very slow fraction of slower-moving molecules. So if we take a look at what this looks like inside of the cell for all the proteins, again, same format as previously with the localizations. We have C1 aggregator that at the 0-hour time points, we get these highly dense foci, but then you still get some proteins that are highly diffusive inside the cell. But then as we start increasing concentration, all of those proteins get recruited to these polar foci here, and you don't get any fast-moving molecules in the cytoplasm, suggesting that once they're in this aggregate, they're not getting out. So it's an irreversible solid aggregate in this case. I also want to note here that the color coding of these trajectories are representative of the diffusion coefficient. So highly mobile trajectories will be in this magenta color, as well as more slower moving molecules will be in this green color. And so when we did look at these three other protein systems for the pop textural linker, you see these highly dynamic proteins in the cytoplasm, and then again, these very slow moving molecules at the poles. And you see that the change between one phase versus the other increasing over time. Interestingly, for this PopTag long linker, which I mentioned is more dynamic than the short linker one, you get a pretty homogeneous distribution of fast trajectories inside of the cell. But as we increase concentration, it's actually quite unexpected that we expected this phase separation effect where you get a highly enriched region at the pole. But what we were not necessarily expecting is that the molecules inside of this enriched region would be moving this fast, almost at the same rate as things outside. And then finally, for mCDB, we get a more traditional or expected results that once we increase the concentration, we start seeing these slow-moving clusters around the cell and end up getting localized to the poles of the cell. And so I've just been showing you scribbles of fast-moving things and slow-moving things inside cells, but we actually quantify the diffusion constants here, and I show you the distributions for mCDB as we're increasing the concentration. You see the emergence of this lower population increasing as we increase concentration. And then these are the results for all these different proteins. So for the C1 aggregator, like I said, we start with a lot of static moving molecules, but some really diffusive, but eventually they all converge to this very static slow state. While with this pop textural linker, again, you see the similar trend of this slow state increasing, suggesting that all these molecules are getting recruited to the membraneless organelles. And then for the long linker, just for consistency, we also fit it to a two-state system, but it still appears to be in one, where rather the overall distribution seems to slow down, which is indicative of the condensing effect. And so in summary here, the slow fraction will increase as we increase the concentration at these different time points. And then if we plot the diffusion constants over these three different conditions for all four proteins, we actually see a very nice trend of dynamic states, where if we pay attention to the slow state here, you can see the significant differences between the mobility of these proteins. So for the C1 protein, you get a diffusion constant that is around our limit of resolution, suggesting that it might be just a static moving molecule, which makes sense if it's an irreversible solid aggregate. For the short linker pub tag, you get something that's slightly faster than that, so quite slow moving, but not super dynamic, which is consistent with our other results. And then finally, for mCDB, you get a much faster state here that is still below what we would consider a freely diffusing molecule in the cell. So we think, we hypothesized that this is liquid-like behavior. So these diffusion coefficients of the slow states correlate well with the hypothesized material states of all these different proteins inside of the cell. And so finally, I've been telling you about these dynamics of single trajectories, where we take a diffusion constant from one trajectory. And so we have plenty of trajectories and get a distribution. However, there are more advanced statistical methods where instead of taking one full trajectory into consideration, we look at the individual steps of the trajectories. And using Bayesian inference, we're able to classify these steps into different mobility states. And so here we have PA, mCherry-McdB at the 4-hour time point. And so we run this Bayesian inference analysis method called no bias, which is some work done by one of my lab mates in the Biteen lab. You see here that our slower state has a diffusion coefficient of around 0.07, which is consistent with the values we were getting earlier, and they're still quite dynamic, even though slow and moving. And this is about 64% of the steps are in this region. And then we get a fast population that's around 1/3 of the total population that is in a diffusion constant that is consistent with freely diffusive proteins in our system. But what's interesting in doing this single step analysis technique is that we're able to determine the transition rates between one state or the other. So for mCDB here, from the slow state to the fast state, you get around 13% transition rate. However, for the fast state to the slow state, you get a slightly higher transition rate, but quite showing this dynamic exchange between the proteins in the slow and the fast state. And this is quite striking when we compare it to this C1 aggregator protein, which I've been telling you so far, forms these irreversible solid-like aggregates. So again, the diffusion constant is over a magnitude slower than mCDB, suggesting that it's behaving more like a solid. It's very quite static in nature. And then at this 4-hour time point, the high concentration, 92% of the steps were in this static state. And even more striking here, we have the transition rates going in and out of this slow state. And we see here that the small percentage of the fast moving molecules have a high probability of getting recruited to this aggregate, while this 3% transition rate out of this slow state suggests that this is an irreversible aggregate that cannot exchange with the contents in the cytoplasm. But again, this is some preliminary data, and we're repeating these experiments and measurements to further validate these very exciting initial results. So like I said, these transition rates suggest more of an exchange from the condensate to the cytoplasm in mCDB, consistent with these characteristics of liquid-like membreless organelles. And so with that, just want to end with some conclusions. So I hope that you can see that mCDB and these pub tag tunable condensation proteins exhibit these liquid-like behavior, but it's not binary, right? As you can see with the contrast between the short linker and the long linker, there is a spectrum of mobility, which may be indicative of a spectrum of material states that these proteins can undertake. So we've been using just standard fluorescence imaging to reveal the cluster formation and the solution showing the reversibility of these organelles, which is consistent with our hypothesis. And then using super resolution to quantitatively get the ratio between the cytoplasmic fraction and the condensed fraction of these two-phase systems, as well as using single molecule tracking to access the motions of these different regions inside of the cell. And so with that, I like to acknowledge my lab, in particular my advisor, Professor Julie Biteen, as well as my lab mate Zuyong Chen, and undergrad Surya Gogineni, who has helped me with data collection in this project, as well as my collaborators, Dr. Yi Huang and Professor Anthony Vecchiarelli, as well as the funding source here. And so with that, I'm happy to take questions.
If anyone has questions in the room, feel free to raise your hand. And if you're online, you can either enter the questions in the chat box or at the bottom right of your Zoom, there's a reactions button. You can raise your hand and we can call on you.
Okay, now I have a question. So when you qualify in the a few slides back, when you qualify and color code your slow and fast track, you use 0.05 and 0.09 as filter. About showing later in your two-state analysis, your slow state is actually above 0.05. So the dot value is not really consistent. So how do you decide those thresholds? Decide like 0.05 and 0.09, and how do you, for example, if we have track that like 0.07 is being between, how do you?
Yeah. So the initial motivation for choosing those values was just to get an idea of where the slow moving trajectories were localized inside of the cell. And so to encompass what generally was the slow population, regardless of the material state of our protein, I chose 0.05 because that seemed to be on the upper end of, or I guess the mean of the upper end of our slow moving molecules. And then 0.09 is usually on the lower end of what we would consider really diffusive proteins. And the reason why I didn't include things in between that was because these are distributions. So I didn't want to overlap between things that might be slowly diffusive, but may be color coded magenta as fast moving molecules. But again, those are arbitrary values that we can change to get a clear understanding of the localization of these different dynamics inside of the cell. Yes.
Going back to the diffusion plot where you sort of categorize solid, gel, liquid.
Yes.
Yeah. So how do you classify those? It just
So these are just hypothesized states based on the diffusion coefficient. So based on in vitro data of PopTag and mCherry-McdB, suggests that these are behaving more like liquids rather than gels in at least some of these cases. And so because they are so near what we would consider really diffusive, the next phase, trying to keep it simple in terms of material state liquid, and then something that's static because it's below our resolution, I would consider that solid. And then anything in between would fall under the spectrum. So just arbitrarily choosing gel-like, but of course, there's many different things that need to be considered there when actually assigning the material states of the protein.
and it's a continuum.
Yeah, exactly. As you can see here, there's quite a nice trend of these diffusion constants demonstrating the dynamics of these proteins.
What's really cool is that when we compare that to what you show in your single particle with squiggly line plots, is that even though you still have fast diffusive long liquor contact, you're still getting liquid phase separation.
Yeah. And so I'm arbitrarily fitting it to a two-state model for consistency, but I assure that if I were to do it to just a one-state model, you get a decrease in trend of diffusion constants, which would be indicative of the confinement due to this phase transition.
Oh, wait. Y'all said there's no questions online, but so there's one. So from Dana, great talk. Thank you. Sorry if I missed this part, but do you have any comment on the biological function of this transitioning state property of mCherry?
Yeah, so these are all hypothesized models for now, but earlier on I mentioned that the internal components of carboxysomes, which are the membraneless organelle, which mCherry-McdB associates with, they're suggested to be forming through this phase transition and actually be quite a liquid-like compartment inside of the cell, in contrast to what's been historically thought due to electron microscopy experiments that undergo these like dehydration and fixing processes, which converge on a crystalline or solid-like nature of these compartments. And so why mCherry-McdB is undergoing this phase transition may have to do with, I guess, more of the kinetics of mCherry-McdB, both recognizing carboxysomes as well as releasing from carboxysomes for, I guess, an active recognition of positioning. And so carboxysomes that are no longer functional may not be recognized by mCDB due to their pH being different, for example. And so they're no longer positioned inside of the cell and just get sent out to the pole, which is actually something that has been shown in the literature not so long ago, that dead carboxysomes get degraded at the poles. So they're not being positioned anymore. So mCDB may be having definitely is serving some function there. Whether the phase transition is responsible for that, we're trying to understand that better. Thank you for your question.
Questions in the room? Give another moment in case anyone online is typing. I think a lot of people have already logged off, but a few still out there.